Oh, it's the right word.
ברוכים הבאים. This is our monthly research seminar. We have decided this year to do it by Zoom. So we usually do it at 7:30 till 9, which is why there was a misunderstanding in the last email. I apologize, but you're here. Thank you. We did anticipate more people, so there may be people still coming in, and also there might not be because people got confused. So we're sorry about that, but we are recording, as we like to do. And I want to welcome you and hope that you'll all pay attention to when we have further ones. It is usually the second Tuesday of the month, which fit right in with when Audrey was here. We're going to do this a little bit less formally than her talk this morning that she gave in psychology. She flipped the order. What she's going to talk about now is behavior phenotyping, and then talk about measuring change and how we do that. But she started with the measurement this morning. We get the phenotyping, which is really much more cogent to the things that we're doing. So it is my amazing privilege to present to you Dr. Audrey Therm, who is a child clinical psychologist and research scientist at Boston Children's Hospital and a member of the Faculty of Child Psychiatry in the Department of Psychology there at Harvard Medical School. She worked for many, many years at the National Institute of Mental Health and there ran a program for rare diseases and looking at the neurodevelopmental and behavioral phenotyping services, watching basically as the definition of autism changed and as research in autism changed. And I think that what we can learn from her is the fact that she focuses on improving diagnostic and cognitive assessment tools using longitudinal studies of autism, so looking through change. And I do think that anything else I can say would probably be personal because I am very honored to also count Dr. Therm as a personal friend. We worked together many, many years ago at the University of Chicago. And then after she finished her training, she was a Lady Davis Fellow here in Israel with me for a year. And now she's back. So I'm not going to say much more because I want her to speak, and there will be time for questions, or if something comes up and you want to ask a question during, she said that's fine. And she will be talking about behavior phenotyping of intellectual disability and autism. And I am going to ask you if you would prefer for me to turn off the overhead lights. I'm going to show you what that looks like. Or if that will put you to sleep. What would you prefer? This? I'm going to be like an eye doctor. What's clearer, this or this?
You guys
decide. Or this? What would you like?
We can start with lights, and if it's
let's start with lights. Because
too much lights.
Compromise.
Okay. Much thank you for joining us.
well, I'll add a personal, and then I will actually talk about my career, starting with our time together for a little bit here, because I think that my experience conveys my message in a lot of ways. But the personal thing that's not on any of these slides is that I'm also married to a Hebrew University graduate, so that I'm just so thrilled to be here today, back here today, and I hope to come back more. I have disclosures that, especially since this is being taped. So as a government employee, I wasn't allowed to do consulting at all, but now I'm starting to, but I haven't actually started, but I am keep putting these disclosures out. And that also, even though I'm not with the government anymore, especially since this is being taped, I want to be conveyed that a lot of the work that I'm talking about comes from the work done that I did at the federal government. And I'm still a special volunteer. So this service, which I'm going to talk about in a minute, came from actually doing autism research. And the autism research that I started with was very lucky to be with Corey in, the initial longest, besides British, longitudinal study of autism, which is still going on. And in that study, really at the time, we were really just still trying to understand what autism was and wasn't, and what the course of autism was over time longitudinally. We weren't actually even trying to figure out what subtypes were so much. We were literally just trying to figure out what it looked like in young children, how stable it was over time, and what it looked like over time. But when I came to NIH and started a study in 2005, 2006, we were interested in asking questions about etiologies and subtypes. And so we replicated that same longitudinal study into a new one, same ages, same idea, but actually at the time, believe it or not, vaccines were the issue, and so was the burgeoning idea of genetics. And fast forward, the findings from that study, that second study, where we did sedated MRIs, we got research approval to sedate children for MRIs and lumbar punctures.
Oh my God.
Yeah, we did, on 67 kids and twice on 31. And those samples still exist at NIH, and they've been used for various things, including looking at folate, which is something that we're talking about again now too. Anyway, the biggest subtypes, and we were looking at regression specifically, the biggest subtype that we came up with after a very expensive, long longitudinal study focusing on etiology was that a bunch of the kids had genetic findings. That's really, whether you had a genetic finding or you didn't, was the subtype. We didn't have enough kids with genetic findings at the time, because we did the genetic testing in like 2011, before exome and genome sequencing, but there were some differences in kids who had genetic findings versus not. But as I was doing that at NIH, trying to just keep in my lane of autism research, I started to be approached by geneticists at NIH. And NIH doesn't have very much pediatric work, but the pediatric work that was happening there was very focused on conditions where they were studying the basic biology of those conditions, and most of those were considered metabolic. But the list, the neurometabolic right there, those geneticists came to me and they said, all the parents are asking about is, does my kid have autism? And we need to measure IQ. These kids are very delayed. A lot of them don't talk. Some of them don't walk. We need to do, neurodevelopmental testing. Can you do it? And I said, sure. I thought, yeah.
Are these kids autistic?
Yeah, I'm going to get to that. Yeah. Some of them already had diagnosis of autism, but most of them did not. But some definitely did, and they a lot of the parents were asking that question: does my kid have autism? Because this was in the very late 90s and beginning to it was in the 2000s, 2010s, and autism was becoming a question for a lot of these kids. To get services at school, especially, I'm sure, like it is here. So I said, sure. So I just took my toolkit that I have, which is a bunch of IQ tests and how we use them in autism and the little tricks that we have and the ADOS and all of our measures, and I just started to apply them to these conditions. And I did see that depending on the condition, maybe a third to a half, depending on the condition, the child could be diagnosed with autism, or at least they failed those measures. But this is where I started to see, number one, sort of the age and amount of regression and the time that course happens in matters a lot. And then also that a lot of these kids have a lot of other medical issues, including sensory impairments like vision and hearing loss, where it becomes almost impossible, if not impossible, to answer the question of whether they have autism. and you may have seen, and I can come back and talk about the paper that I then more recently wrote about differentiating intellectual disability from autism, because a lot of it comes from this work, that a lot of these children were very impaired with intellectual disability, and on top of that had other sensory impairments, and that's when, you know, measures that we have in our toolkit become invalid, like the ADOS. You really, you can't just use a cutoff when kids have mental ages less than 18 months, which you all should know, but then also if hearing and vision impairments are so great, or motor impairments are so great, that you really can't differentiate. So for some of these conditions, we've published papers on percentage with autism that we found, but others we take a more sort of continual approach to looking at those measures, a lot of times because the measures weren't valid in those conditions. But what you could do across all of these conditions is look at adaptive functioning. You could look at cognition, even if you do have to use different measures. And you can start to look over time, which I'll get to in a minute. But some of these conditions were neurodegenerative, which added a whole new complexity to what we were doing, because even though in our understanding of autism, there are kids who regress, you don't see the level of neurodegeneration that I've seen now in some of these kids. So from this, over time, we decided to stop studying autism idiopathically, partly because that group was getting smaller and smaller as genetic testing is coming, but also partly because it didn't seem to be the best use of resources at NIH to be studying idiopathic autism when all these genetic conditions were being studied. So I put my effort towards those conditions and created a service where we were providing clinical consultation or assessments, because a lot of the children that we see either were so sick or they were so focused on their metabolic conditions that they weren't actually getting neurodevelopmental testing at all, and services in the US are not so great, or they were just getting it at school but they weren't getting comprehensive evaluations. So we were providing those evaluations and at the same time doing the phenotyping, and then at the same time, that's what's across, sort of measuring how our measures worked while we were doing that. So we were trying to accomplish all those things at once, which was not so easy. So now I'm going to talk about behavioral phenotyping in the context of all this work, and sort of what I've come to understand about the notion of behavioral phenotyping, where it came from, and what we need to be doing with it now. And those are the themes: heterogeneity, how, and harmonization. Okay, so behavioral phenotyping has probably been in existence for much longer than I am quoting here. But Elizabeth Dykins, her career was at Vanderbilt, she's still there actually, and she really studied Williams syndrome and Prader-Willi syndrome. She did amazing work defining behavioral phenotyping and defining it as a heightened probability that persons with a genetic syndrome will show certain behavioral or developmental sequelae relative to those without the syndrome. So that could be autism, it could be intellectual disability, it could be pica, or irritability, or self-injury, any of these things. But I think it's really important that her definition, I think, still holds very true, is that it's just a heightened probability. It doesn't mean that every child with that condition has those issues, and if they do, they will be in varying degrees, as this figure illustrates. Because I think what we ended up doing is doing a lot of reifying based on early findings that were very much anecdotal, that weren't done with rigorous, quantitative phenotyping in a lot of these conditions. And it really bothers me that, we say that kids with Williams syndrome are hypersocial, or all kids with Prader-Willi, have these feeding problems, when they all don't. A lot of them do, but not all of them do. And again, it's varying degrees, and those things change over development.
Then what does define them as having a Williams syndrome?
That's a great question. Their genetics. Their genetics is what defines them having this Williams syndrome, and I'm going to show you exactly something about that in a minute. I think so it was done, and I show this book. This is Recognizable Patterns of Human Malformation. It's Smith, I think, yeah. And back then, and this is like from the 50s, this book, that's what made them recognizable back then. It was dysmorphia. It was their faces, and body parts, and the way their image was. That's what defined phenotypes back then, really. And then they would try to study specific behaviors. But there was no genetics. There was Down syndrome, and then in I think 60s became Fragile X. There was no genetics really until the 80s, for the most part. So Down syndrome was recognizable. The genes weren't. The genetics was not identified for I think 100 years or something. I mean, it's been known forever, right? In various terms based on the dysmorphology. But then the idea was. These conditions have different faces, and then they have different behaviors, and then the genetics came a lot later. So the questions in the past were, how are these syndromes distinct? We could see there were some distinctions, although they did not do such a great job making distinctions based on facial, or body phenotypes. So then, how were they distinct behaviorally? That was the question, and that was a really good question at the time. When again, there was no genome sequencing that spit out results within 2 weeks of exactly what was going on. It didn't exist. We didn't even know the DNA. But then once they did figure out genetics, we started to ask the question of within a genotype, how are the kids different based on phenotype? And now we're asking the question of even very specifics within genotype. So kids with Down syndrome. There's differences, and then kids with single gene mutations where it's the same gene, like SHAG3 is one of the genes, but there can be many different specific mutations that what can be wrong with the DNA within a genotype, and we're trying to see if there are specific genotype-phenotype correlations there. So these are the two questions that behavioral phenotyping were answering for a very long time. So, but it's all about differences, right? It's all about differences between groups or differences within groups. But it's all about how do these conditions differ. And these are the conditions. So I love that. I think this was from Dykins in 2007, which, isn't that long ago for some of us who are old, but is a lifetime ago for this kind of research. She showed the 10 years between 94 and 2004, how, she thought this was exponential in publications about these conditions. Well, I can tell you, it's 100-fold, right, of publications for all these things now. But also, this was the entire list of what they were doing behavioral phenotyping on at the time. This was it. This was the list. So these were the things that either, some of them, they didn't, Rhett's on here, but we all know that the gene wasn't identified until 99. So we knew that Rett was a condition that had a specific genotype, but we didn't know what it was, right? Okay, does anyone know how long this list is now?
How long what?
How long this list is now? Well,
I thought everybody wants to be on the autism spectrum because it's kind of for social reasons. So, the considerations are more complicated than, if anything, there is such a thing like ground truth. I'm not sure it's related to this list. It's about social benefits from being diagnosed with specific characteristics, isn't it?
Well, I know we're in the social work department.
You could say whatever you want.
So, yeah. I think it depends what lens you're looking at. So, I've been in medical systems at, academic medical centers my whole life. And I think that's why I like the genetics, is because the genes don't lie and they're not based on who's getting services. The genes are the genes, and that those are the conditions that the kids
they're
have.
a very small part of the picture, the single genes, if you're not familiar with the forms of expression that we don't fully understand. So it's all in the genetics, but genetics is not just the genes.
Yeah, but structurally, if you have a mutation in the gene, you have the mutation in the gene. I understand that we don't understand fully the mechanisms of those, but you can at least define conditions based on the gene that has a mutation or the set of genes that do. So that's how the whole genotyping field now has been defined. And this whole genetic first approach to autism is exactly what I'm getting at. That this is now scientifically how grants are being funded and how foundations are being formed and how research is being driven now a lot more, which is what I'll get to in a little bit. And this is not just about autism. This is about the entire behavioral phenotype, the entire way that behavior and other medical systems are expressed, right? So thinking more broadly than just about autism. So the idea back then from behavioral phenotyping research, again, this was when we had a very short list of genotypes, and we were looking at whether or not you could say that there is some behavioral phenotype that's specific to Down syndrome, Prader-Willi, whatever it is, and that would be total specificity. That you could look at a child, you might see different faces or not, but you would see behavior that's distinct, and then you would use that to guide treatment. And cognitive adaptive outcomes of each or other behaviors are not common to those syndromes. I'm now trying to get us back in the mindset of those who were studying this 20, 30 years ago. This was their approach. And they would start to look at genetic conditions and ask these questions and try to use the same tests, the same measurements across the conditions to be answering these questions. They literally did not know because, again, the conditions were just being identified. So, was there total specificity? The conditions were very different. Some had autism, some didn't. Was there absolutely no specificity and everyone's the same? And that's how we've been treating, for instance, idiopathic autism or idiopathic intellectual disability. That's how we've been treating them as these broad entities that are just based on similar behavior profiles. You could ADHD too. We're treating this way, or whether there's this idea of partial specificity, whether there's some overlap in behavioral expression of genotypes, but there is also some distinctions. And I think we all agree that this partial specificity is the case, but we don't know the extent of it because we haven't done such a great job of using measures across the lifespan for all these kids, or knowing what all these conditions are. But in general, I think we agree that there is some sense of partial specificity from what we know right now. But I'll speak to that more. In the extreme approach, and I love this book for existing, but you probably won't see it being a bestseller. But in the extreme approach, we would do what this book did, and this book you can buy it on Amazon. It's saying that there is specificity of these genotypes to the point that there were chapters written about how you would apply ABA to Fragile X versus Down syndrome versus Smith-Magenis. And this book is not that old; it's 2014. So even that 11 years ago, it was thought that you could make a book that would help you, and that you would read the chapter on Prader-Willi and know how you would apply ABA to Prader-Willi. Well, I think that if you were going to do this book now for all the genetic conditions, you would be having 1,000 chapters at least, because that's where we are with genes that are associated with intellectual disability. But I also think that that this book assumes what I said was a little bit maybe oversimplifying that behavioral phenotypes are specific to the point that you would, ignore the other chapters and think that just because you have a child with Williams syndrome that they would meet all that criteria and not have other things going on, right, or change over time. So, I'm sure this book is helpful for again, if you're in a classroom and you've never seen a kid with Williams syndrome before, it was thought that at least you could read this and you might not need to read a whole book on Williams syndrome, but this chapter which gives you some idea from at the time what we knew about the phenotype and apply that. Okay, so now how has the landscape changed? And it keeps on changing. You know, in the last, it's really sort of exponential how much is changing. But now, as I said, this is from 2015. There was about 700 genes at that time. Now we're way past there that are identified to be associated with intellectual disability. So, and you can see, you know, the types of sequencing we're doing. Exome sequencing just started in 2010. Now, whole genome sequencing is the type that's much more frequent. We're way past this. I'm going to show you more. So now we're at, you know, depending on whether it's genotype first or phenotype first, we're at thousands and thousands of genes existing that we know associate with some phenotypes. And depending on how you define it, like I said, we can say very safely that there's 1,000 genes that are associated with it. Some of these genes will have recognizable syndromes, but actually a lot of that work was done when Smith wrote their book. Now, most of the kids we see do not have anything recognizable in their face that at least we can tell. Some, you know, might have some subtle features, but they're not recognizable even by a geneticist. And again, how can we expect a geneticist to memorize 1,000 genes and how that might look? But now it's much more up to just the genetic testing to tell us this. And as I said this morning, as we're doing this, it always starts with one kid. And how I can tell anyone who wants to know later how they finally decide that a gene is what you call pathogenic or you know, causing a disease. They do that based on comparing it to thousands of people who have this testing that are healthy, and then they finally do some clinical work with animals to show that this gene is indeed pathogenic. And then, so then one person gets online and they go on Facebook and they say, now I've been identified with this condition, my kid has X, Y, and Z. Then another person on Facebook comes on, and then 2, and then 4, and then 8, and then enough to form a foundation. And now there are hundreds of foundations now that have genes that are associated with neurodevelopmental conditions that are out there. Some of them are still very rare, and others are, as you can see, you know, thousands of cases. And the problem here is that, and I'll show you in a minute, you need genetic testing in order to identify the cases to then figure it out. So this is just the list that's right now on the Simons Foundation website, which is a foundation that is devoted a lot of funding to research with this perspective of genotype first. First, they did a very large study of over 50,000 kids with autism, where they did free genetic testing in all those individuals, and a lot of those kids were then, the findings were then used to identify a lot of these genes that, again, sort of crowdsourcing with other research made these genes become what they chose as the genes that they were going after to study. So this is a very short list of the 1,000 genes that I'm talking about. On the left here, and I'll show a slide in a minute, are conditions that are not based on one gene. All the letters on the right are one gene. The ones with the numbers on the left are chromosomal abnormalities, so they're conditions that affect not just one gene, but multiple genes on a chromosome, where it's either deletions, where the genes are gone, or duplications, where there's too much of the gene being expressed. And some of these are not so rare, like 16p. There's thousands and thousands out there with it. And others, like I said, are pretty rare still. But I think all of these have several hundred kids identified at this point, some with thousands. So again, for the copy number variants, this is the idea that there are some CNVs that, you know, as you can see, ID is what's at the top, and that's why ID is on the title of my talk, is ID is what is common to all of them, intellectual disability, but some have varying degrees of depression or bipolar or OCD or autism or ADHD. And again, this is from a shorter list of those, but I did just want to talk about these. You don't hear as much about the CNVs or copy number variants as you do about the single gene conditions, because they are not as easily amenable to gene therapy, because there are multiple genes involved. The gene therapy that, if any of you heard me this morning, I talk about, that's with the conditions that just have one gene, because that's what you can target in gene therapy. And anyone who was or wasn't there this morning, this is not just, in theory anymore, and I'll show you. These trials are happening. I don't know, Gal, I might pick on you since you're still here. Are any happening with sites in Israel?
Sorry. I think there was an animal trial that is run in Sheba.
Yeah.
But as far as I know, that's the only one.
Okay. But they're coming.
Yeah, I know.
Yeah. And definitely people from Israel can go to the States or other places and participate in the research. So here it just shows that if you do get genetic testing, what the hit rates are. So if you're someone with autism or intellectual disability, the hit rate it is growing. So the likelihood that you would be found to have one of these genetic conditions is growing because we keep on identifying more of them. And so this changes everything about what we're doing with behavioral phenotyping, because we're not doing what Elizabeth Dykens did of having a short list of 7 conditions. We now have thousands, certainly hundreds that we're looking at. And so, how we're going to sort of group them, how we're going to conceptualize them, all has to change. And yes, for autism and neurodevelopment, we're trying to map this on. So who has more? As I said, it's all about heightened risk, right? We can't say that some of these conditions are autism or not, because none of them are 100% or 0%. And so this is from in 2019, NIMH brought together a group of translational researchers to a workshop to say, okay, let's make a framework for this. And so this is from a paper that was, I think, called Framework for Studying Neuropsychiatric Conditions Using a Genetic Approach. And so we can approach it really broadly, like in the top left, which is kind of that heightened risk approach that I'm talking about. But we do have to consider the level of intellectual disability as a framework for how we're going to consider all the other conditions, because as we know, how you talk about things like anxiety, how you diagnose them is different depending on the level of intellectual disability. So that's why that's on the x-axis there. But it's just showing that there will be a range for all of these conditions, but what that range is and how broad it is will differ, and then that's how you would approach how you consider the condition. On the right is just showing effect sizes for what comes when you look at the whole literature broadly about who has these conditions. So again, ID is underneath, but you can see ASD a lot, and then schizophrenia less. On the left, down here is really important to show that the research that we're producing, and when we look at it in a whole, how much you see of these conditions depends on the way the research is done. So when it's in an individual case study or report, you can get a lot of detail about the conditions because they're writing a lot about them. When you do case series, you're getting less information, and when you do big cohort studies, oftentimes they don't group kids in the way that we do diagnostically, so you're getting less information. And this is when you're trying to get big data on what's going on with these conditions more broadly. And then importantly, individual factors about the child, or when they're diagnosed or how they're diagnosed, is really important with their genetic condition and how you're looking at it. In this case, for sex chromosome aneuploidies, whether they were diagnosed prenatally, and a lot of these conditions can be diagnosed prenatally, and that's a whole issue, in itself, is prenatal testing happening. Whether they were diagnosed prenatally or not made a huge difference in how they were, intervened upon as they were young. And so the prenatal testing group ends up having higher IQ scores than the postnatal testing group. And we think a lot of that is because the kids who are getting genetic testing postnatally are getting it when they have a lot of problems. So they're more likely to be a more impaired group.
Although are their parents more likely to test them prenatally?
Just thinking the same thing.
Parents with higher IQs are more
Well, but
likely
this is the
test
kids,
for that.
not the parents.
Yeah, that's genetic.
And the prenatal testing is actually based on whether there's a concern in utero. It isn't based on
Choice.
Yeah, exactly. Yeah, or older age for this testing. Okay, so it isn't totally the case, but now guidelines for neurologists and geneticists and pediatricians are all coming to the conclusion that that for intellectual disability and or autism, no matter what, there should be genetic testing. And I don't know how that's playing out in Israel right now, but it's not so good in America either. But from geneticists and certainly neurologists who are seeing more and more kids with these genetic conditions, they're saying do genetic testing on everybody with intellectual disability and autism. Do it on everybody, and yeah.
When you speak about exome, is it trio exome or just exome for the child?
Yes,
itself?
it needs to be trio. It does need to be trio, and that's because you need to know if the condition is de novo or not, if it's hereditary or if it's new in the child.
Okay.
But may I say that in some cases it will enough to have a single program. It's
I'm saying what's recommended. It's
Yeah.
not happening. It's not happening all the time. Yeah.
so you know if there are limited
Especially-
resources, a single
Yes.
exome can.
Especially in Israel, we have many programs in relation to other countries, including the most populations in the USA.
Yeah.
So,
But and there are many times for many reasons why the trio can't happen. And what they're doing is you're doing the proban first, and then the trio based on the results of the proban. Because if you find something that's a known autosomal dominant, you don't even need to do the parents.
yeah.
But it's when the findings are ambiguous that you need to. And not only do you need to do the testing, and you know, genome testing has its pluses and minuses, but it certainly gives you the most information. You also need to keep further evaluating, because as I said, today while we've been sitting here, probably a gene has been identified, right? So it's the truth. So and they know this now to go back and reanalyze the data. About every 1 to 3 years. So this is the recommendations, or what is going to be put forth in the recommendations. And I know there's a lot of pushback to this for a lot of reasons, and I'm happy to talk about that. But in general, the reason is because we now at least have clinical trials and a lot of efforts being put towards treatment and considerations for genetic conditions, including prognosis of the likelihood of developing seizures, scoliosis, many other issues that knowing about can be helpful. But it requires kids be diagnosed with the condition, the whole approach that I'm talking about. And so because of that, I've also become involved in research looking at what's happening, at least in the US, and would love to do this more globally. So these are 2 publications from very recently to get Medicaid data, which in the US is the public funded insurance, which is only a fraction of children, unfortunately. It's very hard to get this data, and so these are lagging in time, so that this is up to 2016, and then I think the one on the right is up to 2019. So this is before genome testing, but it includes some exome testing, and you can see here that it is just abysmal, the number of kids with these conditions that are getting the testing, and then this is the types of testing that they were getting. It was just, in 2017, only 3% were getting exome sequencing, which only tells you that we're at the tip of the iceberg of actually diagnosing these conditions, because if kids are not getting the genetic testing, they're not getting the diagnosis. So I just wanted to talk about that, and we can, again, talk about the pros and cons of genetic testing, but this is the rubric that I'm talking about. Okay, so what's happened in the last 15 years is that the number of genes implicated in IBD and their development has exponentially risen. The methods for detecting these conditions have been developed and are becoming more feasible, and they are becoming a lot more financially doable, cheaper. That's the English word. And the number of children receiving the testing is too low, but it has increased a lot. So, now many of these conditions, as I said, don't have recognizable or specific syndrome features. So, and they certainly haven't been studied enough. So, in addition, the mechanisms of a lot of these genetic conditions are being explored with translational research, identifying treatment targets, and by that I mean either small molecules like a drug or gene therapy that might target them. And I talked about Rett syndrome this morning. Angelman's has mentioned these are conditions where there's at least 2 trials in Rett, and I don't know, 6 or 7 in Angelman now that have different modes of action of gene therapy in play. And there are probably, well, actually I have the slide. So this is actually from 2021 of conditions where gene therapy is in play for. And as you can see here, a lot of these were conditions that I happen to that were being studied at NIH, and that's not coincidental. That's why this is happening. So this is all too old now because it says Rett no trials, and there are. So and same with Angelman's. So these were the top hits, and again, only some of these were considered neurodevelopmental, but I will argue from having tested and seen a lot of these kids that they're all neurodevelopmental in the sense that a certain percentage of autism, a huge percentage of ID. And they're neurodevelopmental in the sense that we have to use our neurodevelopmental measurement toolkit for them. Okay, so then that leads to
add something? So
please.
I think one additional thing that is happening that more and more children are being diagnosed. There is less and less cases of variants of unknown significance, like the US is maybe you encounter these. So the more mutations are identified, then it's less common to see these mutations. Oh, okay, I don't know if it's pathogenic or not. This is very important for parents because this result is very disturbing.
Yeah, and that's what I was talking about the sort of in the guidelines that you go back every 1 to 3 years and reanalyze the data because something that was considered a boost 5 years ago it is not anymore. Yeah.
What about pharmacogenetic kits for medications and the ability to tolerate certain medications and so on and so forth? We have, at least in Israel, 2 kinds of them. The neuropharmacogen, and maybe it's different in the US.
You're talking about genetics, pharmacogenetics,
Especially what about the MTHFR, regardless of what Trump and the CDC says and everything, because I do have patients with the variant of a 667 CT.
MTHFR is different from the other pharmacogenomics. In the sense that that's a common variant that we all have or don't have, right? So it has its own research on pharmacogenomics. Mm-hmm. But then there's the pharmacogenomics for these rare genetic conditions, and that is really in its infancy in terms of applying it because you need samples to do that. What I can tell you from being a treatment provider of kids with genetic conditions is anecdotally, we absolutely know that the pharmacogenetics, are there psychiatrists in the room?
She's one.
Oh, okay. I think. Yeah, we know that so many of the medications work very differently.
It's like I have to rewrite the book with every patient.
And we are doing that in Phelan-McDermott syndrome, and it's on our foundation website, an algorithm for Phelan-McDermott syndrome. So, for instance, our go-to for aggression in autism, the things that are approved by the FDA, the antipsychotics, do not work, and they're actually quite damaging. And olanzapine and risperidone. It took me a minute.
You're jet lagged. It's okay.
They can be really, really damaging. SSRIs, which in kids with autism can be activating altogether, can be incredibly activating.
I see tardive dyskinesia at the age of 28. I see.
Yeah. So the unfortunately, and that is the number one reason I would say. I listed other reasons why genetic testing is beneficial, but that is a huge one. Is that and we're seeing massive amounts of catatonia, which I couldn't understand why that was in the DSM 10 years ago, and now I fully understand it, and it's being activated based on some of these medications. So I'm happy to talk to you after. So you're a believer. Okay, so now here's the problem, is that, so we are taking this genotype first approach still. We got to that point, and so we're all writing these papers about specific genetic conditions and doing these natural history studies of specific genetic conditions, totally in silos, different groups doing different things, using different measures, and it's it worked for a while when it was a small enough number of conditions and a large enough sample sizes within them. I will argue that it's just not possible now. And Simons did take the approach that I showed you. The genes that were listed on there is what they are calling Simons Searchlight, which is this group of genes that they are applying broad sort of registry-based data on. They're calling natural history, which is not, but they're collecting data to at least get some information on these conditions. However, they're using the tools from idiopathic autism to do this, and a lot of these conditions, the intellectual disability is too great, or autism is just not the issue, or they're just not the right tools, especially for assessing change, as I'll be talking more about. So I just did my own search. This is just the A, B, and C of that list, and I went through, and so every one of them now, I stopped after half of the A's because I was finding these are just papers that are just either just a few case studies or a small series. They're all doing their own studies of phenotypes in one way or the other, but all using totally different approaches. So, I mean, I'll ask you all, is this doable, and does it make sense for what the field needs now? And so, how do we do this? How do we group the conditions? How do we only do genotype, or do we try to group them either by the on the right is a cartoon of like the networks that the genes express themselves in, because we actually do know quite a bit about how they do that. Do we group them in terms of those kind of networks? Do we group them, as they were before, based on this idea of whether there's a metabolic involvement, neurodegenerative, epilepsy? There's many different ways of grouping them. Or do we just still call them, their own syndromes, which you can argue that these are actually not syndromes, because a syndrome needs to have something that ties it together. And you need to know what that is. And if all we know is there's a gene and there's 20 kids with it, it's actually not a syndrome unless we know something about it. And so I think the term syndrome is probably outdated for this. And I wrote a whole paper about how we just need to call them conditions, which nobody's read. So, how do we do this? And these are just different schemas about, whether we think about them as quantitatively differently or qualitatively differently or both. And we don't have the answer, because until we use systematic approaches to doing this, we don't know. How am I doing with time?
You have another 35 minutes till we end.
Okay, so
you'll end when you're ready for
Okay.
questions.
So this is a slide that I showed this morning because this is where it's the same place as I was talking about. I just talked about different pieces of this earlier. Is that what we need now is to figure out how to measure these conditions in a treatment trial. We don't have time to do 30-year natural history studies of 1,000 conditions. We don't have time, resources, people, or anything to do it. What we need to know is what would be important, meaningful change in a trial, what would be success. But we do need to know something about what the condition looks like, in the natural environment, quote, natural history, to know what might change. But as I said earlier, and I'll keep saying, very little attention is being given to this idea that we need to know what the natural history is. They're saying it. You need natural history data, and you need to know what the endpoints are. But how do we do this? And I don't have the answer. But this is at my new shop in Boston. They made this matrix called Gene Target, where they're saying you can rank conditions, these 1,000 conditions, based on these things. Is the genetics understood? Is early diagnosis typical or helpful? You need early diagnosis. You're going to need that for these mechanism-modifying treatments. So I'm highlighting the blue that are the things that I have to do with. Is, do we understand the natural history, and do we have validated endpoints? That's the N and the E. But then the other ones are there tools to deliver, the treatment to the target tissue at the right time? Is there availability of these safe and effective treatments? It has reversibility in these gene therapies been demonstrated in animal models? Is the gene tolerant to dosage changes? And have ethical principles have been considered? And I would highlight that one that we can weigh in on too. But anyway, it's people from many different disciplines that need to work on different aspects of this. And I think one of my take-homes is that we need to all do it together, and we need to not ignore these natural history and endpoint things. What's happening?
Nothing.
Okay. So how to achieve the goals of behavioral phenotyping necessary for what we would call clinical trial readiness? What is in that matrix? At the very least, we need to have some understanding of natural history through longitudinal data with standardized neurodevelopmental measures. And then, as I talked about this morning, at least the U.S. Federal Drug Administration, what they care about is what they deem important for a treatment to be effective. And I'm not talking about ABA, because that doesn't go through the FDA. I'm talking about drugs or gene therapy, is that a patient has to feel better, function better, or survive better. And in most of these conditions, we can't get to feelings easily with neurodevelopmental conditions. Most of these conditions, we don't have to worry about survival, but some of them we do. So we're left with functioning. But luckily, functioning is something that psychologists and social workers like to think about. And then, again, we don't have to worry about these identifiable phenotypes because genetic testing can do that for us. But we still have to understand what happens. So now, in the last few minutes, I just want to introduce the concept. The field came from the idea of behavioral phenotyping, what the behavior is. But I will argue that what we need is the term developmental phenotyping, is that we need to understand the phenotype developmentally, because what we're trying to intervene upon, as I talked about this morning in these gene therapy trials, is development. Not just one behavior at a time, but what happens over time. I think I've made most of these points, but I'll just take a break for 1 second and read this. I think I've said all these things. That's good. But now I'll focus on the last bullet, which is that, I've talked about that we need to use standardized measures. We need to use standardized, what we call phenotypic ontologies. We need to know that we're actually talking about the same thing when we're talking about things. So, are we talking about depression? Do we all know what that means? Are we talking about emotion regulation? Do we know what that means? And we do not use the same terms in the same way, especially in multidisciplinary. So there's this ontology that's called a human phenotyping ontology. It came from genetics, and it's a way of looking at very specific phenotypes or endophenotypes or features that just uses the same terminology. And it's great for things like capillary, whatever that is, of the eyelid. They did a terrific job with eyelids and body parts and and a lot of things that are more structural. The people who made this, who I've been working with, they did not do such a good job doing this with behavioral phenotypes. So these are the behavioral phenotypes that were in there for autism. They had autism as the behavioral phenotype, which, as we know, is not terribly helpful. So we've been working with them on trying to come up with terms that people would understand and use in a standardized way, and that has not been so easy. So I think I think that's all I'll say about that, except for it's an ongoing effort, and there's some complications on that. But then we also need to change our views of sort of how we model it. And our mentor told us that trajectories are what matters. So that's what I'm going on. And I think it's a good idea. And that we need to, take a developmental approach to these developmental conditions. And there's a lot of theory behind here, but that's the main point. And so these are trajectories. So they're not one time point, and this is really what, and these are way oversimplified, but what we need to know for each condition is, how wide is this, how great are these sort of, each trajectory, and, are there multiple for these conditions? And then we know that there may be subtypes for these genetic conditions that we need to figure out how we're going to call success based on how many subtypes there are, and for each individual child what their trajectory is. So doing this is not easy, and it definitely requires longitudinal work, and it requires us to be systematic. So these are just the conditions that I have studied well enough to know that some of them are, as I said, are certainly considered neurodegenerative. Some of them are thought of as neurodevelopmental, but actually as we learn, and Fuller-McDermott is an example, as we learn more and more and we're seeing adults, we're seeing that there is a neurodegenerative approach, and we think that that's probably underlying a lot of the differences even in response to medications for some of these conditions. Certainly we know the most about Down syndrome and how there's immune involvement and there's involvement in that really is relating to brain changes, and that certainly looks like dementia, and we are seeing hints that is the case for a lot more of these conditions, which again is a reason to diagnose, be proactive, and be able to understand more about prognosis and when to intervene.
Audrey, why is there
Yeah. Well, that's the first paper you and I ever worked on. So this is that little circle in autism is about the regression that we know exists in a certain percentage of kids with early onset. How the condition develops, and that's just an indicator maybe that maybe those kids, and actually we did find this to be the case, that the opposite of what we thought would be the case, which was that kids who regress would have exposure that we're not going to talk about right now that caused the regression. In fact, we are finding that kids who had an early regression are more likely to have a genetic finding.
Right.
And so that's all a big question mark about whether that early regression is actually a sign of neurodegeneration. We haven't followed people with autism even enough in adulthood to know that. But I did want to put out there, because neurodegeneration is just regression that doesn't recover. So the concepts that we've seen in Rett syndrome and in autism with onset need to be looked at a lot more. And the same tools or different tools, but systematic approach needs to happen. I think I've said all this. I just wanted to put these in to make sure I didn't miss anything. But this idea that the phenotypic data are measured in a way that is actually sensitive to change. Again, our diagnostic measures were not developed for this. I'm going to skip through. This slide is just showing you that now in a condition like Fragile X, which was studied with many, many different kinds of treatment trials, now it's being recognized that the treatments need to start earlier, and that's what those dots on the right side are showing you, that there are a lot more happening in younger children, which means that we need to actually be doing a lot better job of understanding the early onset and the sensitive period, and what interventions are happening in kids along the way, especially during the early years, because that's where a lot of the action is going to be for the intervention. So this led us to do work where we're trying to see are just basic milestones, and these are the most basic milestones: sitting, walking, crawling, and onset of language. Are they? Can they be outcomes in themselves? And the first thing we noticed is that compared to typical development, which is all these things are happening by 12, 18 months, So you'd have 100%. In idiopathic autism, for motor delays, you have very, very little. And as you can see here on all these genetic conditions, especially the single gene conditions, you are seeing massive amounts of delay in these early milestones, much, much, much greater than you would ever see in idiopathic autism. So a sign to the clinicians, if you're seeing, and we now have a website that we're working on that shows this, just walking over 18 months itself is a major, major indicator that you're going to have a much higher percentage of a positive genetic finding. And you can see here, you know, that and this is across the board, but and certainly different, but even things like if you're going to intervene early, you know, the age that you walk might be a key factor. And then the language delays are just pervasive, including in autism, as we know, but how delayed they are, you know, is different. I think I'm going to wrap up now, or are we with time? I'm going to skip these. And talk about this.
Some of this stuff looks pretty interesting.
I know. Well, I can tell you later, but I do. How much time do we have?
You have about 10 more minutes
Okay.
to talk.
To talk?
Yeah.
Yeah. I think I'm going to wrap up soon.
Okay. Whatever you want.
So I'll just say that there are, you know, the challenge is how do we harmonize? I would love ideas, but there are, I mentioned just earlier, there are organizations now devoted to doing this in various degrees, but they're mostly using remote data collection, using registries, using, you know, assessments, not necessarily the best assessments for looking at change over time and sensitivity to change, but there are organizations that exist out here to either collect health and behavioral data, do statistical analysis in a harmonized way, sharing importantly, and then, you know, promoting interaction between the patient and family groups, and then focusing on the meaningfulness, which I definitely talked about earlier. And now I'm just gonna show you some findings from a natural history study of creatine transport deficiency that we just published. So again, this is this actual natural history study. Some are funded by NIH or, you know, other or the European Agency for single conditions. But again, I think we've basically run out of money to do this. We're just not going to be able to do it for every condition. But other times, drug companies are actually sponsoring these natural history studies because they have a drug in mind and they know they can't get to a trial unless they've done this work, so they're doing it themselves. So this is a study of creatine transport where this is 50 kids, and we followed them for 2 or 3 years, COVID plus or minus, where we were able to do direct testing. And as you can see here, you know, we tried to do the WASI when we could, which is the Wechsler, you know, regular IQ test, and we thought the Mullen would be a backup, which is, you know, an early developmental test, and it ended up that almost all of the kids had to get the Mullen, which is, you know, a test that was then used out of age range, because the Mullen only goes up to age 5. So this is something that we see all the time, that our kids cannot use the tests that are further age range, which means they can't get standard scores at all. And then, you know, what do we see when we're looking at these kind of trajectories? So here you've got, you've got the IQ tests on the top, and you can see the few that got the WASI obviously did better because we didn't have to use ratio scores or, you know, mental age over chronologic age as their score, because that's what you have to do when you test out of age range. So you can see, you know, their scores going down as they would, and being quite low to begin with. You see more variability in the Vineland or adaptive functioning, but this is what gives you the idea of the actual trajectory. Again, you only have 3 years or 2 years for each individual, so you have to extrapolate. And you can see that the adults are doing a lot worse, as you always do with standard scores in adults. But this does give you a picture to then use if you're going to look at treatment to see at least a little bit of who might do better. And then here you can see the specific subdomains of the Vineland, and I talked about personability scores or gross scale value scores this morning, which are the metrics that actually are unitless but are pitting the child against themselves instead of against the standard scores, and you can see the huge difference in what you're looking at here. On the left are the standard scores, where a massive amount of kids are at the bottom or the floor of the test. This is not going to be very helpful, right, in the treatment trial, where you're just going to continue to see them at the floor of the test, where there is still some floor effects, but a lot less when you use these gross scale values, and you can see the change over time. This is just all the data plotted together, but in general, there actually is not a lot of change over time in these kids, even using the gross scale values, which are basically looking at the skills over time. But these are the kind of scores that you want to be looking at in doing a natural history study, to then be able to maybe use this data. Even if you don't have a randomized control trial, you could use this data as what we call external control data. So you would pit anyone who's in the small group of people in the trial against these, and actually be using this statistically to say whether there's improvement in the children who are in the trial, if that makes sense. Finishing up by saying we absolutely need harmonized measures and approaches. We need to use standardized measures in terminology and definitions. We need to focus on the developmental trajectory, on the developmental phenotype, rather than just one point in time or a specific point in time. And we need to conduct natural history studies in a way that creates clinical trial readiness, and to do that we need to collaborate. So I'm excited to hopefully collaborate more with Hebrew University. And with that,
Okay. You want to add something?
No, I want to stop.
Okay. Questions? You're not.
Thank you so much. So interesting. I don't have enough knowledge or background in genetics to understand everything.
Yeah.
But enough to understand how complex the work is. And my question is actually out of the scope of that talk, so you can definitely keep it to the end if there's more content-related questions. But I was interested if you experienced, and if so, to what degree, and what is your response to it, the huge debate in the autism community, and specifically the autistic community, to any genetic research as a genetic research with the claim that no matter the aim or the original objective is, the result might be like eugenics or use of potential findings to screen and get rid of the autistic genetic.
Yeah, I am so happy. I don't think that's out of the scope of the talk at all. I think that's quite relevant. And I'm happy to have a conversation about it. And that's why I showed this. And I've diverted a lot of my efforts to the studies on focusing on who's getting genetic testing, and especially in the U.S., under-resourced kids that have public insurance or no insurance. It is a huge debate, and it's part of the larger debate that's brought us to this idea of profound autism versus not, right? So I think it's a massive part of what's brought us to this. I have a problem with the term profound autism because a lot of these kids, the phenotype is not autism, intellectual disability is what brings them all together. There's now so much stigma about the term intellectual disability that they're using autism, which I think is highly problematic, because we're denying services to those kids who don't get diagnosed with autism, which is a whole other thing, and we're also not doing justice scientifically, because it's also diverted a lot of the research, the animal models and mouse models, to looking for social phenotypes, and those are the least likely to be able to be changed with a lot of treatments. And not to mention that we don't know how to make a mouse social, so it's very, very problematic. It's also not what the parents care about. The parents do not put social on their top list of things that they want changed. None of them, and I talked about that this morning a little bit. Or repetitive behaviors. Neither of those things are very easily amenable to change, or
So it's like the seizures, the
cognition, communication, seizures, exactly. Sleep, GI,
But
those
that
things. Yeah.
it didn't spare like other genetic projects that also aim to deal with this. I don't know if you are aware. For example, I came from Cambridge when they launched the Spectrum 10K project.
Yeah.
And that was exactly the objective,
Yeah.
to focus on epilepsy and other conditions, and the research was just stopped
Right,
due
right.
to a protest, like a massive protest.
Yeah. I think that I think that both forces are very powerful at this point. I think that now you should stop recording.
Right? Yeah.
1 second. I have to get my